We are doing an enzyme lab. This lab is about looking at factors that affect enzymes. The enzyme we're going to be working with is found in all sorts of organisms, including animals and plants. We're going to get this enzyme catalase from a liver. We can also get the enzyme catalase from other organisms like plants and by grinding their tissue, but here it's in a relatively high concentration. Catalase is an enzyme that works in our metabolic processes to break down hydrogen peroxide, which is harmful, into water and oxygen gas. The moment we take the enzyme out of the liver, so by breaking down the cells, the enzyme will start to break down as well. So we need to keep it cool on ice. I'm going to go through every single step. You'll see that I can do this lab in probably 10 minutes or so. Because you're working with your group, it'll take you a little bit longer, but not that much longer if you are prepared. So hopefully watch this video before you come into the lab, both the person doing it as well as the students at home, because you all have to work together in order to get through this lab. We're doing this lab for 2.5 hours. The first time you're going to do is low tech, which I'll demonstrate. The second time will be high tech, which I'll also demonstrate, and hopefully by the time we put it all together, it'll make a lot of sense. So this first step, I'm going to prepare for the class. So you guys will not be doing this first step. I'm going to take my liver. It's really yucky, so I'm going to put my gloves on here. And I'm going to cut a little piece. This would be helpful if I had a small cutting board, but I don't. And I'm going to do 50 into 100 grams. Okay, so I'm going to take some of my liver here. I'm going to cut it. I'm right-handed, so it's a little harder to do with the left hand. No, I use scissors. I'm going to weigh it. So this here is 42 grams. So 42 grams is good. For you guys, I'll give you an exact measurement tomorrow when I create it in the morning. So that's fresh liver, 42 grams. I'm going to take my glove off now. I'm going to place it in my blender. Done with the balance. I'm going to take 100 mils of water. Graduated cylinder, distilled water, 100 mils. Put it in my blender here. It's going to be a little loud. I'm going to pulse this. Right, so I've pulsed it. I'll do it a little bit longer for you guys, but for me, I only need a tiny little amount. So now I have some liver extract, I guess you want to call it. Some people might consider this a delicacy. I consider it rather disgusting. And now I need to filter it. So I'm going to grab my beaker. And I had some special cheesecloth here, so I can filter it. Get my cheesecloth. So this step here I'm doing for you, you won't actually have to do, but it's good to know every single step of the lab. My cheesecloth. And then normally I have an elastic band or something, but I couldn't find them this morning when I was looking for them. So I'll put this cheesecloth over the mouth of my beaker here. And tape that down or put an elastic band. I'm going to pour my liver extract through. So I want to make sure I'm just getting the liquid part. Okay, so I have plenty. Right. And then I'm going to put this in a small beaker. So here's my pure liver. So I'll wash this. I get another beaker here. And I'm going to put on ice because the moment I take it out of the cell, it's going to break down. Some ice here. So when you guys are putting on ice, you're putting like a 600 ml beaker so you can get ice all around it. I have a smaller beaker because I'm going to do this for a very short time. You guys will be doing it for a little bit longer. So now I have my ice. So now I have my liver. So my liver is good to go. Next, I want to set up my contraption. The thing I'm going to be using to measure the amount of activity the catalase can produce. For that, I need a gas collection tube. Okay? And my gas collection tube is going to need to be filled with water. This is my tray, which is also going to be filled with water. So I'm going to take some water. It's just tap water. Okay, so I got my tap water in here. A bit more. Okay, so this is now filled with tap water. And then I'm going to assemble the rest of my apparatus. So I'm going to get my retort stand, clamp. I need to fill this with water, invert it, and put it into this tray. Nice and tight. Sometimes the retort stands are a little loose, so you need to make sure the retort itself is tight. This is a cylinder. If you put it on your desktop and you don't pay attention, it might roll off and break. So always put this down in a place where it can't roll. I need to fill this with water. I want it completely full, so it's overflowing. And then I'm going to invert it, but I don't want to lose all the water inside of it, so I'm going to use this watch glass. I'm going to flip it over, put it into the water. If I do it well, I'll get no bubbles at the top here. If I do it poorly, I'll get a little bit, but if you end up with bubbles in here, it's not a big deal. Just note where the number starts from. So this is like a graduated cylinder or a giant test tube. So it just so happens that this one here is a little crooked. I'd rather have it straight, but it's the nature of the clamp I chose here. The clamp I chose is not the best one, but that will suffice. So there's a giant test tube. It's graduated, so when I start producing gas, I'll be able to measure that value. So this is my model. This is my system I'm going to be using. Now I have a small little bottle. It's my reaction vessel. And my reaction vessel is where I'm going to react the catalase with the hydrogen peroxide. In order to make this work, let's do this. I'm going to need small little filter discs. And these filter discs are what I'm going to be using to pick up the liver extract. So I've already made these filter discs for you. They're sitting in a beaker inside your containers. So I'm going to do some here for my experiment. So I took some filter paper, put it through my hole punch, This one's particularly absorbent, so it's going to pick up quite a bit of the liver. So I'm done with this. And so now I have my little filter papers. These filter papers are going to be used to pick up the liver extract. Inside my reaction vessel, I believe in the instructions, I've told you to use 5 mils of hydrogen peroxide. So you're going to have a beaker at your station, and it's going to be labeled hydrogen peroxide, H2O2, and then I'll leave you some. And you're going to pour 5 mils in. So that's where you're going to use your small little graduated cylinder. You're going to measure 5 mils. So don't use your graduated pipette for this. Use your graduated cylinder. 5 mils. When you get close to that number, that's when the pipette's perfect to help you get exactly 5. You always want to be consistent. You use 5 for the entire class. We can share data. So I got 5 mils of hydrogen peroxide in here. Okay. In this vessel, I'll be taking a rubber stopper that fits this, and I'll be putting a barrel pipette on the end of this. So here's my rubber stopper. Here's my barrel pipette. Stick it in. Cut that off. Here's my reaction vessel. So I'm going to fill this with 5 mils of hydrogen peroxide, and I'm going to use 4 discs. All right? So what I'm actually going to do is I'm going to do the discs first. If I have cream or anything on my hands, I want to avoid touching the disc because I'll end up getting the cream on the discs, and that will affect my experiment. So here are my 4 discs. I'm separating them. I have a very fine forceps. I dip it into my liver extract, so each of you will have your own beaker on ice at your station. And I want to put it past the neck onto the side. Because it's wet, it will stick. You guys learned about water, and you know that water has this adhesion property. So here's my second one. I always want to be consistent. I want to try to get the same amount of liquid liver extract on each of these discs. Okay, that's 2. You don't want them to overlap each other. You want the full surface area. That's 3. And you want them all in about the same area. You also don't want too much. If you put too much on these, then it will drip to the bottom, and that will modify your experiment as well. That's 4. So I got 4 in here. I'll need my timer. And this is where your partner online will tell you when 30 seconds is elapsed. So I'm going to get my timer here. Stopwatch, so I can see that. So I have it running. And now I have my 5 mils of hydrogen peroxide. I'm going to pour it in. I take my bottle, I rotate it so the liver stuff is at the top. I'm going to pour my hydrogen peroxide, my 5 mils, into the bottle. You might get some bubbling that starts immediately, which is fine. This means that you had a little bit too much of the liver extract that dripped to the bottom. I seal with my lid. I put it underneath the gas collection tube, and then when my partner says ready, I'm going to flip it so that the catalase is in contact with the hydrogen peroxide. And then every 30 seconds, I'm going to watch my timer, I'm going to record the value. So right now, hopefully you guys can see that I'm producing a gas, high oxygen gas. The gas is coming up into the tube, and it's displacing the water. So 30 seconds has elapsed. I'll look at my marker here, and I'll see that I've gone down 22. I tell my partner online, and I keep on going. When I hit the next 30-second mark, I'll measure. Sometimes, depending on what kind of concentration I give you, you might get a lot of bubbles in here. So when you're measuring, you measure to the water line. Ignore the bubbles. The bubbles are incidental. They're not going to be a concern. So you measure to the water line. You find the water line, and so every 30 seconds you tell your partner, and you're going to do this for about 3 to 5 minutes. Okay? Generally, you'll find after about 3 minutes, you're going to get minimal bubbles. So 3 minutes is typically sufficient. Right? But when in doubt, it's okay to go over. When I'm done with this, right, so let's say I'm done. Let's say I've done it for my 3 minutes. So I'm done with this experiment. I'm done with this bottle. I'm working by myself, so it's going to be a little bit more time consuming. So I'm going to give each of you 4 bottles so you can just work your way through this and not have to wait for it to dry. I'm going to take my reaction lid off, put that off to the side, come to my next bottle, and then I'm going to work with 3 discs. So I put 3 discs, I dip them in, put them on the side like I did before. I put in my 5 mils of hydrogen peroxide, seal, get my timer ready, flip, and go. If you are working with this graduating cylinder, with this gas collection tube, and it's mostly empty, you'll have to refill it. But let's say the one with 4 discs, it displaced, let's say 30 mils of water. This is a 100 mil gas collection tube. Then the one with 3 discs is most likely going to do less than 30. And so you'd be safe to just do it again. All right? So you don't have to refill this every single time. You just need to make sure you have enough water in here to displace the amount of gas that you think you're going to collect. All right? So if this is at 60 or 70, I'm probably going to refill it because I'm just not sure. So that's the experiment. That's what we do. That's the first part. In just a moment, I'll demonstrate what the high-tech looks like so that you will be able to jump right from this low-tech version into the high-tech version.